Hi, everyone. Welcome to Microbial Minutes. This is American Society for Microbiology's every few weekly update on what's hot in the microbial sciences. I'm Julie Wolf, the Scientific Communications Specialist for ASM. Today is September 24th, 2018, and we have a whole host of really great topics to talk about. So let's get to it. What important things have happened in the microbial sciences? We are going to discuss a story about Zika and a vaccine that may prove as one way to treat cancer. We're going to look at some studies on probiotics. Are they helpful or not? And do these studies really address that question? We will talk about vaccines, both the expectations for vaccines for neglected tropical diseases like HIV and tuberculosis, as well as the efficacy of the influenza vaccine, the vaccine that we are just getting into the season for, that we should all consider getting now that it is entering flu season. And then finally, we're going to end with some new antibiotics and antibiotic combinations that may help treat some of those drug-resistant infections. So let's dive right in. Our first story is a journal article from ASM's MBio journal titled Treatment of Human Glioblastoma with a Live Attenuated Zika Virus Vaccine Candidate. The take-home message from this study is that an attenuated Zika virus may help fight brain cancer, which is really great, particularly because this form of brain cancer, glioblastoma, is an especially aggressive brain cancer. It originates from malignant or cancerous astrocytes, and it is not metastatic in the sense that it travels to other parts of the body, but it can have some of the cells bury within different parts of the brain itself, making it hard to deal by taking surgical resection and removing the tumor and completely getting rid of all of those cells. This is a cancer that is relatively common, 2 or 3 cases for every 100,000 people in the United States. And you may recognize glioblastoma being in the news. This is what John McCain, senator from Arizona, succumbed to just last month. This is also the same cancer that Teddy Kennedy, the senator from Massachusetts, and Joe Biden's son also passed away from. There's no known risk factors other than age. This isn't something that affects a particular population. And as I mentioned, this is a very aggressive brain cancer. The average survival is under 24 months from the time of diagnosis to succumbing to the disease. We've talked about some different microbes that might be able to specifically target and kill the cancer cells in a previous Microbial Minutes session when we looked at a study from, I think it was JCM. No, not JCM. It was a clinical infectious disease or it was something like that. And it was the polio virus that was engineered to specifically target these cells in glioblastoma patients. Now we're going to talk about a different virus, Zika virus, which you may remember was a big deal about 2 years ago when it emerged onto the scene. And now some scientists are imagining that this could also be something that will specifically target glioblastoma cells. There are several different qualities or characteristics about this virus that make it particularly a good candidate to target brain cancer. One is that it does have the ability to transfer into the brain. So it's something that can cross that blood-brain barrier without necessarily having to apply it directly into the brain itself when patients present with brain cancer. But also, it has it preferentially causes disease in brains that are still developing, in the fetal brain of mothers who are carrying fetuses, and the brains are still developing, whereas in adults, it does not seem to cause any type of symptomatic disease, at least that's related to the brain infection itself. And so there was a live attenuated, so even less able to cause disease, Zika virus strain that has been characterized by this same group, as well as worked on by others. And this study looks at the ability of this live attenuated strain, which was developed as a vaccine to prevent the virulent strain from causing disease, but now looking at it and its ability to treat cancer. And so what the researchers did was to take glioblastoma cells and introduce those into mice. And these are specially engineered glioblastoma cells so that they are fluorescent. They can be detected in the mice using whole body scanners, as you see here. The fluorescence is detected. And they treated mice either with mock treatment, so probably giving them a PBS solution or something that is a sham treatment, or they treated them with this live attenuated vaccine. And you can see that the cells take place or take root in the brains of these mice. And when they are not treated with the Zika virus, that they are able to implant and grow, increasing their fluorescence over the course of a month. But that these mice that were treated with the Zika virus do not have that same gross growth as you see by day 28. There is a slight increase, but it's not to the same levels. However, this is not necessarily something that will completely cure the mice or potential patients should this move into the clinic. If you look at the survival curves, you can see that the mice that are treated with mock therapy succumb quite quickly to this infection after about 28 days. So where these tumors in the whole body scan are very fluorescent and showing a lot of growth, this is around the time that they succumb to that cancer as well. Those tumors within the mice that are treated do not grow at the same rate, but eventually do grow large enough to cause death within those mice. And you can see that death curve shown here. It does increase the amount of time that the mice survive, and that in and of itself may be a positive result, lengthening the life expectancy of this very virulent cancer, this very aggressive cancer, so that patients can live beyond the average 24 months, of course, is something that would be progress. But this also may be something that could treat patients in combination with other, for example, with surgery or with other engineered microbes, such as that poliovirus. And so this was picked up by a few different outlets. I can see Newsweek highlighted the fact that this had just been the cancer that John McCain had succumbed to, and that this is a potential new treatment for that particular cancer. It was picked up by Popular Science, where Justin Lathia, who's actually not related to this study group, but has worked on similar types of research problems, pointed out that these findings that were published in MBio were done in the absence of an intact immune system. So those mice that had the glioblastoma cells, they did not have their fully functioning immune system. And Lathia continues, it's unclear how the immune system would be altered, potentially in a positive manner, to reduce tumor growth. And that's an important thing to point out. A lot of these engineered microbes act with a 1-2 punch. They not only directly infect and kill the cancer cells, but they also kill them in a way that activates the immune system to seek out other tumor cells and kill those even without infection. And those that 1-2 punch is something that's summarized in our Microbial Sciences blog, which you can find on asm.org/blogs, or we have a link down below. And you can read a summary of some of these different the engineered microbes that are being used to treat glioblastoma specifically. The poliovirus, which we've covered before, this now Zika virus, and there's another virus, an adenovirus, which has been engineered to potentially treat glioblastoma patients. And if you're wondering where Zika went, because we are talking about Zika now when we haven't really seen it in the news for a number of months, there's another post that you might find interesting, Where Did Zika Go? And it talks about how both herd immunity and public health measures have worked to make Zika perhaps less of an issue, at least for the near future. All right, we're going to move on to our next topic, and that is looking at probiotics. And there are two studies that were published back to back by the same authors, the same groups. You can see here, a lot of the names are on both papers. And this was in the journal Cell. We're going to talk about both of these, but the take-home message is that probiotics and their efficacy depends on many variables, which may seem a little glib. I think that everybody who's listening to this conversation or this presentation knows that probiotics and their efficacy depends on many things, but we'll get into it and you can let us know what you think. So the first of these studies that we're going to talk about is the post-antibiotic gut mucosal microbiome reconstitution is impaired by probiotics and improved by autologous fecal microbiota transfer, or FMT. So antibiotics, when one takes those systemically in particular, have major effects on the gut microbes, even though that antibiotic is taken in order to target a specific microbe that's causing an infection. There's many bystander microbes that are also killed by these antibiotics. And that's something that I learned in speaking with Colleen Kraft, who does a lot of these fecal microbiota transfers. And so we'll include a link if you're interested in hearing her work. But she mentioned that it takes about 6 months after a course of antibiotics for the gut to reconstitute its microbiome so that it looks the same as before taking those antibiotics. And so this study wanted to ask how we could possibly improve recovery after antibiotics. And they wanted to compare using a fecal microbiota transfer from the host's own baseline microbes, so taking samples before the antibiotic treatment and giving those back to the same host, versus using probiotics, which is a very broad category, as we'll talk about in just a second. But in this case, is a particular mix of many different species that are known to have specific good interactions and good outcomes for host health, including things such as bifidobacteria and lactobacilli. And they also wanted to ask in this second study, how do individuals react to probiotics? Since after all, each microbiome really is unique to the individual based on one's exercise, diet, sleep habits, etc. So let's look at the first study, this one that compares the probiotics and the fecal microbiota transfer. That's some very lovely graphics, which help to discuss their scheme, their different experimental schemes. And they did this study both in mice and in humans, but of course, doing it in humans has more relevance for human health. So we're going to highlight those experiments. They took patients before treatment with antibiotics, and in some, they collected stool in order to prepare fecal microbiota for a transfer afterwards. They then treated all 3 of these groups with antibiotics and then gave them a secondary treatment to help them recover from that antibiotic treatment. In one case, the control case, they did nothing. There was no subsequent treatment, and those they called the spontaneous recovery patients. In some, they gave them a mix of different probiotics, and in some, they gave these patients a fecal microbiota transfer from their baseline microbiota, so here in the gray. And then they looked at the ability of these different probiotic species, as shown here along the y-axis, to colonize different parts of the intestine. And this was very unusual. This is not something that people usually do in different type of microbiome studies. Often we're looking solely at stool, which mostly represents the large colon right before the stool leaves the body. But here they actually use endoscopes in order to explore both the lumen, the tube part of the contents of the intestine, as well as the mucosal contents. So they would take scrapings and examine what types of microbes were in those. And you can see that in the spontaneous, we'll start with the spontaneous because this is how most of us actually recover from antibiotics. You can see that some of these species already do exist in various parts of the intestine. And here we're going in order, basically from the very small intestine to the large intestine as they travels down the gastrointestinal pathway. And you can see with the probiotics treated group that these species were able to colonize in different parts, not necessarily in the large colon, which is the part that we would be testing by stool, but throughout the different parts of the intestine, you can see that there's differential colonization by these different probiotic species. And that those that were treated with that fecal microbiota transfer, they actually don't have these same probiotic species as are seen in the patients that were given the probiotics. However, we're not, our gut microbiota are not solely the probiotic species. We have a very complex or a community of different microorganisms that make up our different microbiota. And so here you can see in aggregate, this is how the naive or the baseline levels of gut microbiota look, with a number of different species represented, red being a more highly colonizing and green being less found than the basal levels. And after antibiotic treatment, it completely reverses. So most of these species are no longer found, right? And so after a month after that subsequent treatment, either with spontaneous probiotic mediated or FMT mediated recovery of that microbiota, can see that there are clear patterns just by looking at the numbers here. Those patients that had spontaneous recovery, they go back kind of similar to this naive or baseline. It's not entirely the same. You can see that there's a number of species that are missing here in the dark boxes, and that when patients were given fecal microbiota transfer, some of their own microbes from before antibiotic treatment, there's a little bit more recovery. We're actually missing a few species here, but it is overall generally a little more red than when patients do the spontaneous recovery. But that the probiotic treatment actually goes to complete green. It still looks as though the patients are under antibiotic treatment without the recovery of their baseline levels of these different members of the microbiota. And you can look at this either by species, as they do here. And this graph down here shows basically the function of the microbiota. So, of course, there are many, many, many different species, and some of them do redundant function, making certain metabolic byproducts that can confer health benefits to the host. Maybe it might come from multiple species. So if we're looking here at function, you can see that same red and green pattern that we see when we're looking at the actual species themselves. So the other question that they wanted to look at was how different individuals respond to these different probiotic or to the same probiotic treatment. And so in this second study titled Personalized Gut Mucosal Colonization Resistance to Empiric Probiotics is Associated with Unique Host and Microbiome Features, it was a slightly different scheme for their experiments. Again, they looked at the baseline levels of what microbes were there. And then they treated some patients with probiotics and some with placebo. So these patients would still have some sort of treatment and followed up to look at what microbes were there, both at the individual level and at the group level within the different treatment groups. And they looked both during this probiotic treatment and then after about a month, the patients were no longer given these probiotics, and just looking at what the effect was on this gut happened about 2 months after that. So if we look at these groups in aggregate and looking just here, we're looking just at the stool, not looking throughout the entire intestine. If we look in this treatment period, there's a clear difference in the microbiota. We can see that the probiotics load, the number of different species that are related to the treatment is much higher in the treatment group than in the placebo group, as one might expect. But that if we look in this follow-up period, it's very similar, that those probiotics do not necessarily fill a niche or implant to become part of the commensal microbiota of those different treated individuals. But that even this treatment differs, or even this response after treatment differs quite a bit. So we can categorize this by the different participants, as the scientists do down here in the x-axis, and then looking again at the different regions of the intestine, can see that those implantation takes place quite different. So here we're looking at the implantation of the presence of the different probiotic species, and you can see that there are a large group of patients or subjects that the scientists call permissive, meaning that they did take up those probiotics and the probiotic species continue to be found within that intestine, whereas the resistant group, or the patients or subjects that the scientists called resistant, really don't look any different than the placebo-treated group. So these resistant subjects likely wouldn't see those same type of health benefits as the permissive subjects, given that the probiotics are something that will benefit human health. And you can represent it in this heat map as shown here, or you can show it looking at relative amounts as they do in this graph here. So what are the conclusions from these studies? If you look at the scientists involved in the studies, you can see that they are happy to promote these and let people know that these different studies are out, and that the lead scientist here, Aaron Elenov, is quoted also in a piece from The Guardian, in which he states, "This tells us that the currently used paradigm of one-size-fits-all probiotic preparation and treatment should be replaced by a tailored therapy which harnesses science and measurement and technology. And this was something that this finding that these probiotics did not necessarily help all individuals equally was not surprising to a lot in many people within the scientific community. Although these studies were shared by Eric Topol, who is shared his he's kind of a expert in a lot of different biotechnology and health medicine technologies. And so he shares a lot of things that are related to both microbiota and microbiology, as well as other types of health breakthroughs, health technology breakthroughs. However, there are several different caveats, and the International Scientific Association of Probiotics and Prebiotics, that has a vested interest in using probiotics, did write a rebuttal to this to these pair of studies, and they highlighted that these were headlines, the news headlines, which were driven by sensationalism and not the data. They pointed out that there are a number of different issues with these different studies. Some of these are pointed out also by members of the scientific community on Twitter, and they include things like there were no clinical endpoints measured. So the scientists of these studies did not look at particular symptoms, so whether or not the patients were more sensitive to particular allergens or whether they responded differently to vaccines. All of these things that we've talked about on Microbial Minutes in the past, there were no actual clinical endpoints. It was simply looking at whether or not those different probiotics embedded and whether they stayed with the patients and affected the outcome of antibiotic treatment. However, probiotics are not always meant to be something that embeds or colonizes a patient or a subject. They can often be something that is taken transiently because as they pass through the intestine in that lumen, their metabolism may still benefit the person who's taking those probiotics and not necessarily confer benefit after the probiotic regimen has finished. And so that all of these were different points that the ISAPP made in their rebuttal. We'll link to that if you're interested in reading it. And of course, the ISAPP has some obvious skin in the game in promoting the benefits of probiotics and prebiotics. They also pointed out that the lead authors of these pieces they may also have some bias due to their involvement with a personalized microbiome company that they did not disclose, which of course is not too uncommon. There are many scientists who will capitalize on their findings and parlay that into expertise in applications for human health. But the failure to disclose this on their actual studies is something that this group found to be a lapse in judgment. So this was, as I mentioned, discussed in a number of different places on Twitter, just like that study on the use of ethanol in hand washing or in hospitals, that there were people on both sides pointing out positive remarks from one group or another. And I thought this comparison of probiotics to different breeds of dog was actually quite apt because probiotics do they serve particular purposes, and it is important to make sure to tailor the probiotic that one is taking to the purpose. And the analogy here is buying a guard dog and choosing a Chihuahua. It's not going to be as effective as a Rottweiler. All right, we're going to move on to the next study. And this is something that was covered in Gates Open Research. The title is Developing New Health Technologies for Neglected Diseases, a Pipeline Portfolio Review and Cost Model. So the take-home message from this is that global health costs are higher than current world budget. It's a look into the economics of developing some of the products that can treat neglected diseases or diseases that maybe aren't so neglected, but will require quite a bit more research in order to bring products to market. So what this group did was to look at a number of different roadblocks, so such as the lack of candidate products, such as the lack of facilities or the cost of facilities to develop these products, and the cost to take these things through the pipeline, through the number of clinical studies that it would take in order to develop either a vaccine, something that would prevent the spread of these diseases, or a treatment, something that could be given to individuals once they are infected. They pointed out that funding for neglected diseases has dropped In the last 5 to 6 years, we now spend about $3 billion a year. This is from nonprofits, from government agencies, from different health organizations. The exception that they pointed out was Ebola, which during the West African Ebola outbreak there was an uptick in funding to immediately treat Ebola. And of course, we're seeing the benefits of that now with the current DRC outbreak, where there is several different vaccines and treatments that are being used to try and stem that outbreak. So this group wanted to investigate. They identified 665 candidates for 35 diseases, and they applied these to a financial model. And they were able to use this model to estimate the costs that it would take to move candidates through the pipeline and predict the likelihood of the launch of different products. They outlined the different diseases that they looked at. They were looking at 35 different diseases. They had a list of the ones that had the most products, and the products could be biologic, diagnostic. There were vaccines and different types of vaccines, simple vaccines, complex vaccines, or perhaps even an unprecedented vaccine, something that hasn't been used previously. And so they highlighted all the number of different products that they were looking at, as well as the cost that those different types of products take through different preclinical and clinical testing phases. So they were able to incorporate 538 of these products into their model, and they estimated that to get these 538 products through the model, it would take $16.3 billion, which is obviously more than the $3 billion a year that we are currently spending. The bulk of this money would be necessary in the next 5 years, with about $4.5 billion a year needed in order to move all 538 candidate products. And from this, of course, some products are going to fail, either in the preclinical or in one of the clinical phase trials. Some of them will prove ineffective, or some will not prove more effective than current measures. And so what they predicted is that this would eventually lead to approximately 128 product launches, but that even if under these ideal circumstances, with 16.3 billion spent total, 4.5 billion in the next 5 years, this would not be enough to get the complex new chemical entities and things such as a highly effective HIV, TB, or malaria vaccine to market, not only to market, but also at a price point that makes it viable to give to the underserved countries, or the places where they might not have the same resources to deploy this to the populations at greatest risk. So this was highlighted in the New York Times, where the lead author, Gavin Yamey, said that this really points out how donors are cutting back on funding at a time when we should be stepping on the gas, and we may be missing a valuable opportunity to take some of these different candidate products to market because people are donating less money to the research of these particular diseases and the ways that we might be able to stop their spread. So that bodes poorly, unfortunately, for a lot of the vaccines that we've discussed even previously here on Microbial Minutes. But we are going to now move and talk about a different type of vaccine, one that is already out on the market, and that is the annual influenza vaccine. This is a study from Clinical Infectious Diseases titled Intraseasonal Waning of Influenza Vaccine Effectiveness. And the take-home message from this is that protection from influenza vaccine quickly wanes. And so, as I mentioned at the opening here, it is now influenza season, or it's around the time of year when people start going to the pharmacy or their healthcare provider to get an influenza shot. And this is a shot that we have to get every year for a couple of reasons. One is that the different strains of influenza that are predicted to be circulating change. And so we need to make sure that our immune systems have seen the proper strains so that we are protected against whatever strains might be passing within the population. But also the immunity that the flu shot builds is not long lasting. We don't get good memory from the influenza shot. And even if the same strains were circulating every year, it would benefit us to receive an annual influenza vaccine, which is one of the reasons why people are working so hard to generate a universal vaccine that not only fights all different influenza strains, but also confers much longer immunity. And so, what this group wanted to look at is how long is the influenza vaccine effective? And so, to do that, they looked at individuals who received an influenza vaccination between 2010 and 2017, and then later tested positive for influenza. So, the people who were able to become vaccinated, but then ended up contracting influenza anyways, because the vaccine is not 100% effective. And what they found was that the risk of flu increases after a few weeks of vaccination. So after 42 weeks, it takes about 2 weeks for the influenza vaccine to become effective. And so the maximum efficacy was seen between 14 and 42 weeks. And that after 42 weeks, this starts to decrease quite a bit. And that correlates with an increase in risk for that vaccinated individual to contract influenza. And so what you can see here on the right-hand side is a table where the days since vaccination. Oh, days. I have weeks written down here. I'm sorry. This is only 42 days, which is a month and a half or so. So the days since vaccination are listed here in this column, and the odds ratio, which is the probability of contracting influenza, is shown in this column here. And you can see that it increases as the number of days since vaccination have occurred, whereas using RSV, which is respiratory syncytial virus vaccination, the odds ratio does not increase. There is no correlation between RSV vaccination and the risk of influenza, of course. And so this was picked up by a number of different outlets. It was picked up by CIDRAP, the University of Minnesota Center for Infectious Disease. Something and protection. And this was also picked up by a commentary in the same journal, Clinical Infectious Disease, where the author of that commentary, Mark Lipsitch, said that my informal sense of the literature is that the suggestion is strong enough that if people could reliably get vaccinated the week or two before flu season starts, they'd be better protected. And so there's been a bit of talk of this on social media and Twitter, discussing whether or not it would be better to change the recommendations for when one should get the flu shot. Normally, it's recommended that everyone be protected by the end of October. So within the next month and a half, we should all get our annual flu shot. But if the protection starts to wane after 42 days, that means that by the time that influenza hits at its hardest, which is generally the mid-December through January, then we would not necessarily have as great protection as if we had received it more recently. And the director of CIDRAP, Mike Osterholm, commented within the Minneapolis Star Tribune, saying that we have to adjust the practice of medicine to the science of medicine, even if that will lead to more people being vaccinated at the same time rather than spreading it out over a month and a half. Maybe we should all do it in the last week of October, and Osterholm is for changing the recommendations based on that. And so this was, as I mentioned, discussed in several places on social media, and you can easily find those discussions, and we'll put links below the show as well. If you're interested in learning more about influenza, we've had several really great conversations with people who study both how the influenza vaccine is made, with Stacy Schultz-Cherry, and the differences between females and males and their susceptibility and their reaction to influenza vaccination with Sabra Klein. We'll also add links to those. All right, we're going to talk about our last subject, which is antibiotics. We're going to talk about two different studies quickly. One is going to be from Nature. This one is titled Optimized Arylomycins Are a New Class of Gram-Negative Antibiotics. And the take-home message here is that scientists have developed a new drug to fight bacterial infections. And really, this is not just a new drug. This is a new drug class, which is a very exciting development, because most of the new drugs that we are developing as antibiotics are really iterations or slightly chemically modified versions of drugs that are already available. This hits an entirely new target that has not yet been introduced into the human population, and thus there should not be a lot of intrinsic resistance within the bacteria that are causing diseases right now. And so antibiotic resistance is a growing problem. I probably don't even need to mention that to people who are listening to a Microbial Minutes session. Gram-negative pathogens are particularly difficult to treat for a number of reasons. A lot of the resistance is passed around in gram-negative pathogens, things such as Klebsiella pneumonia or E. coli. Additionally, even if they aren't resistant already, they are inherently difficult to treat because they have two membranes that protect the inside of the cell. And that means that a drug to get to the inside needs to pass through those two different layers instead of a single layer as seen in the gram-positive pathogens. So this group identified aurelomycins, misspelled here, sorry about that. And this targets a signal peptidase, which is on the outside of the bacterial membrane and is something that would not necessarily need to pass through those two membranes in order to be effective. By itself, the aurelomycins are not particularly potent. They're produced, I believe, from the Streptomyces group, but they haven't been found to be effective enough in order to pursue as a human drug by themselves. So what this group did, and this is from Genentech, and we'll talk about that in just a second, but this group modified aurelomycin and made a compound that they named G0775. You can see the native natural compound aurelomycin up here in this box, and then in the lower part of the box is the G0775. They modified several different chemical characteristics that give it greater affinity and avoid some of the natural resistance found within certain types of bacteria. And you can see that the minimal inhibitory concentration, the mix here for different gram-negative bacteria, actually decrease quite a bit. So you need a lot less of this G0775 to inhibit the bacterial growth than for the natural aurelomycin itself, which is great. But of course, this is only useful if it's able to treat infections. And so this group tested in mice. They tested a number of different gram-negative bacteria. They infected the mice. This is in a thigh muscle infection model. And they saw this is the initial amount that is infected into each of those different mice from the gram-negative. And then they gave them vehicle, or again, this is a sham treatment, and of course the bacteria will be able to replicate if they aren't given an antibiotic, and all of them increase in that case. But when treated with this aurelomycin, the G0775, you can see that the number of colony-forming units, the number of viable bacteria, decreased well below what that inoculum level was. So this is something that this drug looks promising in treating a number of different gram-negative bacteria. They did a dose curve in a lung infection using not just Klebsiella pneumonia, which is what they used here, but an MDR isolate, so something that is already resistant to most of the different classes of antibiotics that are effective. And they saw that it takes a low dose, so this is increasing and then decreasing doses. They can see with 2 mg per kg here that they are able to bring down the CFU per lung, the number of bacteria per lung, and inhibit growth, and that if they go beyond that, then they're able to actually decrease the number beyond what the inoculum is, as shown here. And this led to a greater survival as well. As you can see, the highest dose, this 5 mg per kg, is able to rescue all of the animals, whereas at that 2 mg per kg, although the bacteria are inhibited from increasing, there might not be enough of an inhibition to allow the immune system to fight the remaining bacteria. And while it lengthens their life, some of them do eventually succumb to that infection. And so this shows a lot of promise. This is from Genentech, which is very interesting because a number of companies have closed down their efforts on research of new antibiotics. And while Genentech did get this from acquiring another company, it's promising that this industry, this industrial giant, has developed this new potential class of antibiotics. So one of the researchers said that the aurelomycin natural products were like diamonds in the rough. They needed to strip the scaffold down to its core and then systematically polish all of the edges, revealing in the end its potential for remarkable potency. And this is something that people are very excited about. And in fact, Genentech has produced a YouTube video about this product. It's about 3 minutes long. It's the lead authors of the study discussing their research in a very understandable way. So I recommend that you watch this if you're interested in the discovery of this particular product, and we'll link to that below as well. And so our final paper that we're going to discuss is Nature publication in their Systems, Biology, and Applications journal that looks at the combination of different drugs. So this is a journal article titled Prevalence and Patterns of Higher Order Drug Interactions in Escherichia coli, E. coli. And the take home message is that antibiotic combos may lengthen efficacy of current drugs. So, in the absence of new drugs like the arylomycins that we just discussed, we may be able to use some of the drugs in combination with one another, especially if they have different drug targets. If one drug targets DNA synthesis and another drug targets cell wall synthesis, they may be able to work in tandem. And so, as I mentioned, antibiotic resistance is a growing problem. I'm sure you all realize that. And that resistance in other infectious diseases, such as HIV, has been countered by multiple drug cocktails. In HIV, the magic number was 3. That was the number necessary to prevent the adaptation of the virus to be resistant to the drugs that were being used. And so this group wanted to ask, how might antibiotic drug cocktails increase drug potency against multi-drug resistant bacteria? And so they systematically took 2, 3, 4, and 5 drug combinations, and they looked at how this affected the ability to inhibit E. coli. And they found both that the increasing number of drugs used correlated with increasing frequency of net synergy, meaning that the effect was greater than they would have been able to predict based on the independent activity of these drugs. So 1 and 1, 1 plus 1 in this case equals 3 rather than 2. But they also saw that increasing the number of drugs could increase the frequency of emergent antagonism, where one drug might counteract or somehow negate the efficacy of another. And so these different drug combinations were summarized in the Venn diagrams here, showing synergy or antagonism based on the different cocktails that they used. Now, of course, this is very promising in certain aspects. As one of the senior authors said to Science Daily, people may think they know how drug combinations will interact, but they really don't. And this type of study will help medical professionals to apply different drugs in combination for extremely aggressive and recalcitrant infections. And this was shared by Battle Superdrugs, one of the great Twitter accounts that covers a lot of drug resistance issues, saying that this seems like good news. I think the big caveat that everyone has here is that, of course, this is going to affect more than simply the infectious microbe, just as we were talking about with the probiotic study. Any antibiotics that one takes will affect all of the different microbiota that are part of our microbiomes. And it's most likely that these will be deployed in situations where, a single drug is not able to counteract some sort of infection. And so that's going to do it for today's Microbial Minutes. Oh, so there's a question. This is great. So the question is, can we select novel probiotics from the fecal microbiota transfer positive microbiome? That's possible. They were really used as 2 different aims in that study. So using probiotics versus using fecal microbiota transfer. I guess if there are probiotics strains that are related or more closely mimic the complex activity of the fecal microbiota themselves, that may be something that would be useful. It also depends, of course, on how they are packaged and what their infectious doses are, right? So, for example, we didn't really cover it here, but the FDA has opened up their guidelines for defining what it constitutes a probiotic. Should we use CFUs? Should we use grams per per dose or something like that? And so they've been thinking about maybe relooking at what the effective dose is. Right now, people use CFUs, that's colony forming units, but that might not be the best way to even measure what an effective dose of a probiotic is. So as that develops, it's possible that we'd be able to use a probiotic that would act to encourage the natural microbiota, the same way that the fecal microbiota are basically probiotics in the sense that they are what you want to be taking up that space anyways, and doing all the different functions that our complex microbiomes do as well. I'm not sure if that answers the question, but that's the most that I could say at this moment, because quite honestly, it's a very complex issue. Another question. This is great. Are recombinant vaccines useful in this case? And I am going to guess that this has to do with the influenza vaccine question. And almost all of our vaccines are, in some case, genetically manipulated or recombinant. Many of them have. So I guess a lot of the subunit vaccines in particular are those that have simply one little area of the genome, of the influenza genome that is used. But I don't know if that would be the answer moving forward. I know a lot of people are looking at totally different molecules than the different hemagglutinin and the neuraminidase. People are looking at the M protein or other parts of the influenza virion that might be able to confer greater protection, and whether that ends up being something that can be expressed exogenously using recombinant technologies, or whether it needs to be a live killed influenza, not live, but a killed vaccine is something that I think will depend on future studies and what molecule it ultimately is that confers the best protection. Hopefully that answers your question, but thanks for asking. All right, so thanks everyone for listening to Microbial Minutes. We've discussed a number of things that are happening in the microbial sciences: the attenuated Zika virus that may fight brain cancer, probiotic efficacy that depends on many variables, as we discussed, the global health costs, which are higher than the current world budget to solve a lot of the necessary treatments, vaccines and treatments to treat different neglected tropical diseases. We talked about how the protection from the influenza vaccine wanes quickly after the initial inoculation, and how scientists are developing or have developed a new drug to fight bacterial infections, and that combinations of antibiotics may help fight those as well. Now, I'm going to be traveling quite a bit in the next couple of weeks, so if you have any stories that you want covered in the Microbial Minutes in the next every few weeks, it'll probably be 3 weeks, 2 or 3 weeks until our next session. Leave a little note here, and we will make sure to take a look at what you think are the hot things that have happened in the microbial sciences. Thanks everyone for listening, and we'll talk with you next time.